Good afternoon. So I'm Paweł Szczerbiak from Jagiellonian University in Poland, and we'll be talking about relationships in the microbial protein universe. So we recently submitted our preprint to bioRxiv. And so the main goal of our initiative, called Microbiome Immunity Project, is to understand the role played by different bacteria in human gut microbiome. And this is a very complex problem. So in my talk, I will focus entirely on structural part and structural annotations. But yesterday at Microbiome Cozy, Mary Maranga covered the functional part. So if you missed that talk, there will be a recording after the conference. So in the first stage of our project, we focused on kind of cross-section of microbes that live on Earth. So we took 1,000 free reference microbial genomes from Genomic Encyclopedia of Bacteria and Archaea, which we then clustered. And as a result, we've got roughly 2.4 million of raw protein sequences. And then we split them into domains and generated multiple sequence alignments. And for the processing, we used only those sequences with deep enough MSA, which resulted in roughly 300,000 of representative domains. For structure prediction, we used two completely different approaches, namely Rosetta and DMP-Fold. At that time, AlphaFold wasn't available, but once it appeared, we also used it for to verify the most important part of our data set, namely the periodic novel folds. Of course, we also imposed model quality assessment criteria. And as a result, we've got roughly 200,000 of high quality predictions, which we call MIP structures or MIP database. In our analysis, we focus on comparison between different predictions, predictive novel folds, protein structure space visualization, sequence structure function relationships, functions, annotations, and public access database. For function prediction, we used a tool called DeepFry that was developed last year by Grigorevich and collaborators, which is a graph convolutional network with language model. As an input, you provide sequence of structure. As an output, you have a vector of annotations that are linked to specific Go terms. And so that topic was covered by Mary yesterday. And so our data set, so a little bit about statistics. So our database is in some way complementary to the AlphaFold database because, first of all, we focused on microbial sequences entirely, and they are quite short. So if you take a look at the histogram to the left, so our sequences are between 40 and 200 amino acids, so they are pretty short. But thanks to that, they are vast majority of them are single domains. So that's quite important. And to the right, we have a density plot that shows, so on the x-axis, we have agreement between those DMP-Fold predictions and for two different data sets, namely for raw predictions and for curated, namely those are structures for which we imposed model quality assessment criteria. And it's expected that the agreement is better in the second case. For further analysis, we use the curated dataset. When it comes to predictive novel folds, we use the widely used and set forward, in some sense, definition that those are structures with TM score below some predefined threshold with respect to the known fold space. And by default, we use 0.5 for the threshold. So first, we superimposed all of our domains of our structures against CAF superfamilies, and those that survived, namely with maximum TM score below 0.5, were then superimposed against PDB-90. And as a final criterion, we also wanted to have predictions that agree with each other. So the agreement between the NP fault and the predictions should be higher or equal than 0.5. And so we've got 452 structures of this kind, which we then clustered structurally into 161 putative novel faults, putative novel faults clusters. And we also did alpha fault verification. And surprisingly, the average TM score between Rosetta and alpha fault and DMP fault and alpha fault is higher than the agreement between Rosetta and DMP fault, which means that alpha fault predictions are somewhere in between Rosetta and DMP fault, which is quite interesting. So let me show you some examples. So here we have Rosetta alpha fault and DMP fault predictions on the top and the closest PDB homolog at the bottom. So the TM score between the predictions, between all predictions, is pretty high, it's around 0.8, and also the predicted structure is different from the closest PDB homolog, because those are parallel beta strands, kind of beta barrel, and it's also this is a representative structure from the largest novel fault clusters that comprises 87 similar structures. In this example, the agreement is a bit slower, about around 0.5, 0.6, because of this probably disordered blue alpha helix that was predicted differently by all three methods. But apart from that, just of the structure, the core is predicted almost the same way by all three methods. We also created a protein structure space visualization using quite simple approach, I would say. So for each structure, we created a graph representation based on contact map. Then we computed graphlet counts, on which we then performed dimension reduction using two widely used techniques, namely UMAP and PCA. So this is not a big surprise that the structure space is continuous. It was proven many times in many papers by many authors. So both approaches are able to distinguish between CAF classes. And when it comes to relative contact order, which roughly speaking is a measure that tells us how densely packed are our structures, there is a clear gradient, which also correlates with CAF classes. Namely, on average, beta classes are more, let's say, dense or compact as compared to alpha classes. And also, we annotate our structures when it comes to transmembranes. So also, again, it's not surprising that most of our alpha transmembranes are located in the region where we have more alpha-like structures. I mean, this is not trivial because in some sense it's not trivial because our annotations were generated based on sequence, not structure. This is structural representation here. And this is probably the most interesting plot. So we wanted to know where are our PTA novel faults in the space, and so we found out that they are quite evenly spread across the whole space, but mostly in the alpha beta regions. And also, which is another quite obvious thing, that they are not in the region where there are very, very small proteins, namely here and here. And so last important topic I would like to mention about is sequence structure function relationships. So if you randomly pick structures from a protein data bank, you can get this kind of plot. So sequence identity versus structural similarity. So above roughly 30% of sequence identity, you can get this kind of correlation. Our data set by construction is different because our sequences are not similar, not very similar. So we don't have many similar structures out there. There are some, but not so many. So our data set on average is different to the PDB. And it also holds when it comes to sequence function relationship. But in this case, we also found a more similar, functionally similar structures. As functional similarity was computed as cosine similarity between deep vectors. And so this is probably the most interesting, namely structure function relationship. So the most obvious scenarios are to the left, noted by green circles, namely when we have very similar structures and very similar functions, and very different structures and very different functions. But in our analysis, we focus on something different, something opposite, namely we wanted to analyze structures which are very different functionally, and reversely, structures that are very different but represent similar functions. So let me show you some examples. So here we focus on a specific Go term, namely carbohydrate binding, and superimposed structurally all structures that were annotated with high probability by deep fry to having this function. And interestingly, the largest cluster here is the putative novel fold cluster that I showed you before. And we also found some awkward structure that is probably an outlier. And we also analyze this from a different perspective, namely we compared all of, so we focused on specific structural clusters and computed functional similarity for them, which is visible here as heat maps, and wanted to analyze two different scenarios, namely to the left, where we have a different function that is represented by similar structural motif. And for instance, in this case, we have hydrolase activity and actin binding that is realized by similar structural motifs. And to the right, this is opposite. So we have same function, namely transferase activity that is represented by different structural motifs. And our structures and metadata are available on Zenodo, and there are 3 cases on how you can search the database, namely you can start from sequence, structure, or function, and then infer corresponding annotations. So to sum up, we've generated and extensively analyzed roughly 200,000 of protein models for microbial sequences. We focused on comparison between different predictions, novel false identification, annotations, structure space visualization, and sequence structure function relationships. And our database is complementary to the AlphaFold database due to sequence origin, sequence length, and percentage of single domain structures. I would like to thank all of my collaborators, and thank you very much for your attention. Do you have any questions for Paweł?
So, thanks for the talk. Couple of questions. I'm surprised that you are getting a length of no more than 200 amino acids for these sequences. For microbes, the average length would be about 300. So there's something going on there.
Yeah, that's a very good point. I mean, we imposed a cutoff on sequence length. So it was created by, this is how our database was constructed. So because we wanted to focus more on domains and not on whole genes. So those are just structural domains.
Okay, so the distribution is not representative.
Probably, yeah, probably it's not.
Okay, all right. And then another question was, you showed that cosine similarity value set.
Yep.
That's supposed to be -1 to 1. We're looking only at 0 to 1.
Yeah, but the scores are positive. So as a result, you have a positive
You're saying you have a cosine similarity. Are you taking an absolute value of that?
No, because if you compute a dot product of positive values, you are getting positive value. So it's between 0 and 1.
Okay, maybe I don't understand your scoring mechanism. Okay, but and final. Oh, you have one. So final question for me would be, you have a prediction that the AlphaFold prediction that you're talking about is in the middle between Rosetta Fold and the damp thing,
The
right?
DP fold.
Right, and I'm not sure how to interpret that. So I understood your prediction to include both Rosetta Fold and this damp situation. I'm not familiar with that. And AlphaFold in the middle doesn't really make much sense to me how that would occur. You know what I mean? So why would that be the case if what you're predicting is reality?
Yeah, so we didn't analyze it so deeply why AlphaFold predictions are somewhere in between Rosetta and DP fold. For our perspective, it's desired because once we decide to to run AlphaFold on all of our structures, the whole picture shouldn't change much because the agreement between AlphaFold and our predictions shouldn't be very different. I mean, it should be even better than between Rosetta and DMP fold.
Okay.
Good question. I'm curious, how many of these 160 novel folds are widespread in many different organisms, and how many are
Yeah,
just
that's a very good question. So far we haven't analyzed taxonomy. So this is the thing we would like to analyze next.
But you could have a count, like how many of these 160 reappear in your database many times among those 1,000 genomes?
Yeah, so we probably we'll take a look at that because we haven't yet.
All right, cool. And we also have a question from the online audience, Chenging Zhang. In the PDB, most protein pairs with sequence identity over 0.4 have a TM score of greater than 0.5. This is not true for your curated data set. Why do you think that?
I'm not sure if I understand it correctly. Because we don't have sequence, I mean, don't have many, if any, I mean, many structures with sequence identity above 0.4 in our data set, like in this right plot here.